Hello everyone, welcome back to another class on regeneration biology. So in today's class, we will try to understand about induced pluripotency and the roles of IPFs in general during tissue and also Hox gene and the homeotic transformation during tissues regeneration and also importance of Hox genes and during homeotic transformation. So the second part, Hox genes and homeotic transformation, will be minimally represented in today's class because we will address that much later. But we'll give more priority to the induced pluripotency and its implications in regenerative medicine. So the pluripotency and induced nuclear reprogramming in vertebrates is a very well established or well explored research area. You may have heard about different types of animal cloning, like you can clone an animal. You may have heard, many of you would have seen the movie Jurassic Park, which is made use of this so-called concept of animal cloning. And also you would have heard about the sheep, first mammal to be cloned is Dolly. So, but the same concept, you can use it not just for creating an animal, rather you can use it for making stem cells from basically to related to that organism. But these days we have moved further ahead and we will see them one by one. So induced pluripotency refers to a process where differentiated adult cells are reprogrammed to pluripotent state. So one option is to have the embryo of that particular species, which has got lots of ethical concern. And other option is just to have the nucleus from say from your skin cell or something, take the nucleus and put it into a nucleated embryo of the same species and then allow to grow. And then now the embryonic cells that are formed, it is not a embryonic cells. First the embryo forms in the group of cells, say morula, blastula, like that. Inner cell mass of blastula, you can collect it and culture it, which is nothing but your stem cell. This is one approach, but still it questions the ethics that you made an animal and you did not allow to become an embryo. It could have become an embryo. And now you are killing that blastula. So that is basically technically a genocide, or even an abortion is a genocide. So ideally that is not the correct way. So the accepted way is, take a cell, say you pluck your hair from your skin, it has got in the bottom of that hair there are cells which is easy, like you will be losing hairs every now and then, or you are losing your skin, or you can take your buccal swab, which when you are doing tongue cleaning also you lose some cells. Such kind of cells nobody cares much, and that cell you collect it, culture it in a petri dish, and you convert them into stem cell, your own stem cell. Not too much of ethical concern. Why? Because that cells are never going to be an embryo in any ways. It is your stem cell which is there at the time of your birth. It is there in your cord blood cell, etc. So that is the benefit, or that is the interesting aspect of induced pluripotent cell. So you are not pushing your cell into an embryonic stage, or you are not making a clone of yourself. You are rather making your own skin cell, your own fibroblast cell into stem cell and using it for your research. So that is the whole concept of induced pluripotency. That is why we have discussed about Yamanaka factors, Thomson factors, etc. And pluripotent state resembling the embryonic stem cell by expressing specific transcription factor, opening up the possibilities for regenerative medicine and disease modeling. So why you need stem cell? To fix a damaged portion or damaged tissue in your body. For that, to make that tissue, because that tissue is no more there in your body, that is why you are suffering. So who can make that tissue? Your stem cells can make that tissue. So who will give you the stem cell? You have to make that stem cell. From where? From your skin cell. So that is the whole concept. So induced pluripotent cell. What is pluripotency? Pluripotent cells have the ability to differentiate into any cell type within the body, a characteristic that is previously thought exclusive to the embryonic stem cell. Now, pluripotent stem cell also have the characteristics of an embryonic stem cell, except that embryonic stem cell, or the embryonic stem cell while present in an embryo, can give rise to an organism. pluripotent stem cell will never give rise to an organism. Induced pluripotent stem cells, in short form called as iPSCs, they are derived from somatic cells like skin or blood cells, like I mentioned just now, that have been genetically reprogrammed into a pluripotent state. It was your cell and it is your cell, except that its genetic nature is being changed. Commonly known iPSC generation is via Yamanaka factors. The discovery of iPSCs was made possible by identifying a set of transcription factors such as OSKM. They are OCT4, SOX2, KLF4 and CMYC. In short form, they are the Yamanaka factors that when expressed in differentiated cell such as skin cell, fibroblast, etc. can induce pluripotency. So these genes when expressed, they can make a differentiated cell into stem cell. So let us see about induced pluripotent stem cells. So induced pluripotency is started with a cell from the body. This is the body and one cell is plugged. And then you are doing genetic reprogramming. And certain genes such as Yamanaka factors need to be expressed and you end up getting induced pluripotent cell and behaves like an embryonic stem cell. And this also culturing has to, it require, or what are the requirements to grow an embryonic stem cell is needed for this pluripotent cells also. Culture iPSC in the lab, you need a feeder layer, you need a leukemia inhibiting factor, etc. And they can be pushed into differentiation into neurons, muscles, or fibroblast itself, or maybe some other specialized gland cells, etc. All possible types of specialized cells can be pushed into or made from this embryonic or the so-called iPS cells, which is mimicking the embryonic stem cell. The advantage is no need for embryos. That is the greatest advantage. And another advantage is there is no ethical concern associated with that. Embryonic pluripotent stem cell, iPS cells, like you can see here, a cell from the body, you convert into pluripotent by genetic reprogramming. And this is a picture shown in a stepwise manner. This is fibroblast and you end up getting embryonic stem cell like pluripotent cell, which can be pushed into differentiation into cartilage, neural tissue, muscle, whatnot. You can make them into specific cell types. So what are the applications of iPS? Why someone will make iPS? What was the purpose? Some we already told you, it's for regenerative medicine. Let us learn more specifically. iPSCs can be differentiated into specific cell types to replace damaged or diseased tissues, offering the potential for treating conditions like Parkinson's, Alzheimer's and spinal cord injuries. Parkinson's, people have less dopaminergic neuron being produced from the substantia nigra of your brain. Because of this, they will have uncontrolled muscle movement. You may have seen sometimes aged people, their hand may be shaking like this. It will be shaking like this. Not you are shivering with the fear, not this way. Like this, it will be moving like this. They are not having good control of your hand. This is mainly because, not that any old person whose hand is moving like, don't picture them as a Parkinson's patient. It needs diagnosis. So in general, their dopaminergic neurons are not producing adequate amount of dopamine, or the dopaminergic neuron number is depleted. So dopamine is very important, and that can also lead to depression, etc. And Alzheimer's, it's because of the neuronal damage. Your short-term memory is lost faster and your old long-term memories are staying lost much later. And then spinal cord injury because of road accident, etc. We have also seen some spinal cord injury, regeneration, etc. in the previous class. But understand, the absence of specific type of, say, dopaminergic neuron, or some of this Alzheimer's case, some specific type of neurons, when they are affected, you can supply stem cells, or you can supply differentiated neurons on that affected area, so that now you have more dopamine, or you have more neuronal communications, etc. So this is the approach. That is why you need stem cell. Disease modeling, another application. iPS is derived from patients with the specific diseases, can be used to study the underlying mechanism of these diseases in a controlled environment. Say a person is having some disease and it is due to a gene mutation and you have no idea what is happening. So you can take that person's cell from the body and make an iPSC out of it, and you can see how that cell is behaving. or you can correct it into a normal cell and see how it is supposed to behave from that particular person's cell. Or maybe because of this mutation, this particular cell is not able to produce an enzyme. Then what you can do is, in that patient, sometimes you cannot replace a cell type. It may not be in a place where you can replace the cell type, but you understood the etiology, you understood what is missing. So like small kid, very small baby is there in your house, say 3 months old, 4-month old baby. It cannot speak, it is crying. Crying can be because of various reasons. Sometimes if they get sleep, they will cry. If they are getting scared, they will cry. If they are hungry, they will cry. If they have got stomach pain, they will cry. So you can't ask. If it is a grown-up person, you can ask, why are you crying, what is your problem? Same way, if a cell has got a mutation, why some activities are not happening, that may not be simply direct, because the mutation may not be in the gene at all. Mutation may be in a non-coding region. So how will you decipher it? So understanding this mechanism, or you have the stem cell and you differentiate them into different types of cell and see, okay, which cell type is affected, going to be affected, can we tweak the scenario so medicines can be designed for that patient. So that is the whole idea in disease modeling. And drug discovery, iPSCs can screen potential drugs and assess their efficacy and safety before clinical trials, because iPSCs have a lot in common to that of a entire embryo. Although it is not an embryo, it has got similarities or characteristic features to that of an embryo. So they can be screened. If some drug is toxic, or some drug is accelerating the cell proliferation, or some drug is favoring a particular cell type being formed from the stem cell, they all can be categorized. Sometimes it may be anti-cancer treatment, etc. It can be beneficial. And personalized medicine, iPSCs made from a patient-derived cells can be used to create disease models or for cell-based therapies, offering the potential for personalized treatment. Like many medicine, allopathic or any Ayurvedic or homeopathic, any medicine you take it, a given medicine will not work in 10 different people. That's why you would have seen in the time of COVID also, doctors normally give a cocktail of medicine. For some people it clicks and they survive. For other people, that medicine doesn't click. It's nothing wrong with the medicine. Medicine was fine, treatment regime was fine, but somehow it was not giving them the desired result as they got it in another patient. So the patient specific medicine is a necessity. Which medicine works for whom? So that has to be tailor-made. Just like if a dress is made for you, your best fitting dress, another person, it may not fit the way it fits you. So maybe it may not be a serious thing for gents who wear loose dress, but for ladies who care too much about the fitness, so they are very particular. So you can understand, a one person's body fitting may not fit other person. Same way, the drugs also, not every drug, not that paracetamol or some painkiller, some specific disease drugs may not give as expected way, may not give the result as expected in one person. So maybe slight tweaking of dosage, maybe slight tweaking of a drug's nature is necessary, especially it has huge implication in psychiatric treatment, etc. So another point we should understand is about transdifferentiation. Transdifferentiation, certain stem cell types can differentiate into cell types that is seen in organs or tissues other than those expected from cells predicted lineage. Say for example, some families you can see, grandfather is doctor, son is doctor, grandson is doctor, great grandson is doctor. So if one more kid is born, you will think, oh, this person also going to be doctor. So this is the assumption about the lineage. But suddenly in the family, one person became a social worker. His father, grandfather, mother, they are not social worker. From the doctoral lineage, you got a social worker from that family. Same way, if a muscle specific lineage or neuronal specific lineage, when they give rise to another cell type which is not expected from that lineage, then we usually refer to them as transdifferentiation. This reported phenomenon of transdifferentiation is very important and useful in the regenerative medicine. Why? Say I want my kidney cells to be formed, and I am not interested in making iPS cell from my skin cell because it's very costly or something. I took bone marrow stem cell I have bone marrow stem cells available in me. Now I am telling this bone marrow stem cell, instead of making blood cell, can you make a kidney cell? So this is the whole concept of transdifferentiation. Example, brain stem cell. One example is brain stem cells can differentiate into blood cells, or blood forming cells can differentiate into cardiac muscle cells. These are some examples. Stem cells at home, that is the environment, the niche. There is a popular saying, right? When you are in Rome, do as Romans do. We have heard about that. Or rather, I can say other way around. If you are in Rome, you will automatically do as what Romans do. This is the much better. If you are a sensible person, you will do as what Romans do. It's like if you go for a function, or if you go for a Western world where people are wearing formal dress in a different way than Indian formal dress. Indian formal dress is different, and Western world formal dress is different. Both are formal dress. But if you are in India, you will wear Indian formal dress. When you are in US or other European countries, you will wear that country's formal dress. Same way, stem cells, based on which tissue they are in, they behave accordingly. For example, if I take iPS and put into kidney, they will not make liver cells there. They will try to make a kidney-like cell. They look at the surroundings. So stem cells have specific niches. Niche is basically the micro environment around stem cells that provide support and signals. Both are needed, support and signal. Signal means chemical signals regulating self-renewal and differentiation. So stem cells should be able to make a copy of themselves, and they should be able to differentiate when needed according to what is needed. Not that randomly I have a pool of stem cell and keep making randomly today neuron, tomorrow muscle, day after tomorrow liver cell. It cannot work that way. It can make a given type of cell based on the signal coming from the microenvironment. So stem cells are there, they can influence the niche, and they get influenced by the niche. So they need to have direct contact. Like we have discussed about the feeder layer in a petri dish. You need to have, and leukemia inhibitory factor to be present in maintaining the pluripotent stem cell as pluripotent stem cell. And lot of soluble factors. Direct contact is one feature that comes into picture, and soluble factors released from the microenvironment, and also intermediate cell. The niche cell will guide another cell to influence the stem cell in a particular manner. It is like making a phone call to a person who is sitting far away from you versus sending a person with a message to convey to that person. So that is what you are seeing. So in either way, the stem cells are too much responsive to the immediate neighborhood, which we refer to as microenvironment or niche. So stem cells, when we use for tissue engineering, we need to follow certain steps. One thing is isolation. Whichever tissue, whichever stem cell you are dealing with, you need to isolate them, and you have to characterize them, and you have to expand them, and then you should learn how to differentiate them. So isolate individual stem cell population from an adult stem cell or any part of the body. Ensure that cells retain their functionality and potential to differentiate. You don't want a dead cell isolated from a person. You want healthy cell. And then characterize and track stem cell population during characterization, because you need to be sure that I took this stem cell. I did not take one random stem cell. And ensure that the cells are transplant ready, in the sense they are ready for culturing, they are ready for putting into a petri dish and expansion in number. Culture stem cell lines into a stable, multi or pluripotent state based on what stem cell you harvested to start with, free from mutations, and of sufficient quantity. You need to have a good number when you are expanding. You cannot do a stem cell transplantation with the 5 cells. You need whatever it takes, or whatever target tissue you are going to push into. It should have that much good quality number, thousands, millions, like that. Enable economical expansion to make cell therapy a reality. Say this expansion requires some $10 million or 100 crore rupee. No patient will be able to afford. So that is one person has to, or a therapeutic approach should consider that it should be viable, and it should allow people to handle that economic burden behind it. And then control and activate stem cell to differentiation to desired lineages. I have a stem cell, but it will differentiate only into neuron, not into any other cell types. Then basically whatever effort I put in is waste. Although I claim it as a IPS, or I made an IPS, which is very nice. It's like, I'm a singer, but I know only one song. I'm a great singer of one song. But after that song is done, then I don't know what to do. So this we don't want in a stem cell. And functionally active differentiated cells. Now I differentiated, but then it's not doing the job what a cell is supposed to do. I made a neuron, but it's a dummy piece. Just like in some dining tables you would have seen, right? You have apple, banana, or other types of fruits, very bright colored, nice colored, but you can't eat them because they are made of wax. They are for beauty purpose they have put it. So they will satisfy your eyes and it will fill the void. You don't want a cell made like that. You want a cell that is functional. You don't want a dummy piece. So one has to make sure that it is perfectly functional cell is being made. So what are the challenges in stem cell research? It is uncertain in that the human embryonic stem cells in vitro can give rise to all different cell types of the adult body. Although it can give rise to 10 different or 20 different or 50 different, but I am interested not in that 50. I am interested in that 51st type of cell which the stem cell is not able to give rise to. Then stem cell has got some lacunae. It is not perfect. So this is what one has to take care of. It is unknown if stem cells cultured in vitro apart from the embryo. In embryo, stem cells are there, but they are in the embryo. They are not in a petri dish. Will function as the cells do when they are part of developing embryo? This is a big question. Will they do as they are supposed to do? Like you are very comfortable in your house, dining room or living room. But if I drop you in the moon without oxygen, too much radiation, will you be as comfortable? Because environment can matter. So can the stem cell do the same job? So simple appearance is not good enough. Stem cell development or proliferation must be controlled once placed into patients. Now I put stem cell which can give rise to any cell type and can proliferate everything. What if it become cancer? One has to be bothered about that. Possibility of rejection of stem cell transplant as foreign tissues is very high. So when you dump, because stem cells can have too much of MHC class 1 expression, and sometimes this class 1 expression beyond normalcy. Normally the class 1 MHC class 1 proteins are less in differentiated cell, class 2 are more in differentiated cell. So all of a sudden your immune system is saying class 1 expressing cells out there, and the body immune system is not facing that much of stem cell around, and it is vulnerable for immunological attack. So chances of rejection, tissue, although it is made from your own body, but it is from a differentiated cell. Stem cell is not common in your body. Your body saw stem cell during your embryonic development. So contamination by viruses, bacteria, fungi and mycoplasma is possible. So because you grew them in petri dish, you cultured them. Sometimes epigenetic changes also possible in those genotype. Maybe some genes will not get expressed at all. And during this process, maybe some fungus, mycoplasma, etc. are there inbuilt in that cell type. Once you transplant, then you not only you cured the disease, you created a new disease also in the patient. So these are all the things, the challenges that stem cell is facing. And some challenges and future direction. Safety concerns, which we already told you, because many of these Yamanaka factors you express using viral vectors. Using viral vectors to deliver reprogramming factors such as Yamanaka factors can raise safety concerns, and researchers are exploring alternative methods for reprogramming such as chemical reprogramming. Use some drugs. No need to put any gene, just to put some drugs so that they will get reprogrammed. Efficiency and reproducibility. The efficiency and reproducibility, like I gave my skin stem cell and gave to a researcher and told, okay, please make my iPSC and give me. And I gave 100 cells. He still, all died. I cannot give me. So I cannot keep supplying kilograms of cells for that purpose. So I can give only X amount of cell. So the efficiency is a major concern. If the efficiency is not good, it is not a good strategy. Like if you are planning to cook some food, if I make 1000 dosa, then only one will be edible one. 999 has to be discarded. Then I am not a good cook. So epigenetic memory, iPSCs may retain some epigenetic memory from their original cell state, from where they came, that is skin cell or something, affecting their differentiation potential. Although you are putting Yamanaka factors, they may not forget. Like, old memories die hard, there is a saying. So immunogenicity, iPSC is derived from one individual may be rejected by another, which is normal, raising concerns about using iPSC derived cells for transplantation to another. Self-transplantation is allowed, and also ethical considerations. Development of iPSCs has reduced the need for embryonic stem cell and addressing some ethical concerns associated with embryonic research. So if anyone publishes embryonic research data, people may ask, oh, but this has no relevance because people are doing this, the iPSCs. So what is the point of this kind of research? So these are all some concerns. Now we will move on to Hox genes and homeotic transformation. Hox genes are a family of homeotic genes because these Hox genes are necessary for differentiation or even the formation of a whole embryo itself. They are crucial for establishing body plan. Body plan basically means you have only one nose, you have only 2 ears, you have 2 eyes, you have only 2 hands, you have only 2 legs. Why can't you have 3 hands? Why can't you have 5 hands? Instead of 1 nose, why don't you have 3 nose? Now these are all fixed because of the homeotic transformation happening in right time. So during development, and mutations on any of these genes during development can lead to homeotic transformation, where one body part develops into another. Like in the place of my nose, now I have a hand. Or in the place of nose, I have got an ear, not nose. In the place of ear, I have got 2 eyes, something like that. Homeotic transformation is a phenomenon in the developmental biology, is the alteration of one body part into another, often caused by mutations or misexpression of homeotic genes. These genes are basically Hox genes, which are crucial for establishing body plan and segment identity. So homeotic transformation, by definition, the homeosis is a homeotic transformation, is the developmental process where one body structure develops into a structure normally found in a different location or a different segment. Mechanism, how does this mechanism? This transformation is often triggered by mutations or altered expression of homeotic genes, also known as Hox genes. These genes are master regulators that control the development of specific body segments and structures. And for example, some examples include Drosophila. A classic example that has been worked out in Drosophila is the transformation of antennae. Drosophila has got sensory organ, which is 2 are present, antennae into legs. In the place of antennae, or in something equivalent, I can tell, in the place of nose, you have got 1 leg, something like that. Because antennae is a sense organ for Drosophila, and that has been appearing in the fruit flies. It means not naturally, you can make it by homeotic transformation due to mutations in a specific homeotic genes. There are different odd skipped, even skipped, different types of genes are there. They are all Hox genes. And Drosophila has got a segmented body pattern. Even our body is segmented. You may not agree, but actually we have segmented body pattern. That is why in some anterior segment you end up getting shoulder, you have pectoral girdle, you have pelvic girdle, etc. Not that you have pectoral or pelvic girdle right in your belly region. It is this girdle is here and not above your head, or head is in the anterior portion. So this kind of identity is because of the segmentation in your body. And anurans, the vitamin A treatment during tail regeneration in anuran tadpoles in the amphibians can lead to homeotic transformation of tails into limbs. In the place of tails, you end up getting a leg. All you did is you treated them with vitamin A. And in vertebrates, homeotic transformations of vertebrae have been observed in murine embryos that are exposed to teratogenic doses of retinoic acid, leading to posterior transformation along the body axis. That means you end up getting legs in by shoulder, or you end up getting hands in the pelvic girdle, etc. So this kind of positional identity is maintained by Hox genes, and they have huge implication in the development of an entire organism. And these Hox genes are responded by the stem cells. So there are I am not going into the developmental biology, but understand Hox genes decides how an organ is being formed from the stem cell during the embryology. We'll study more about regeneration biology in the next class. Thank you.